Hey guys, we're back for lecture 10, which is clinical applications of neurotech, and sorry that this has taken such a while to record and just create, but we've all been busy with our own things, and we hope you guys have all been safe and well during this time. But yeah, let's just jump right into lecture. So here's the outline of what this lecture is going to look like. We're going to start with a simple intro and then go into a few techniques and then a quick review. So the first question I'd like you guys to think about when we're looking at neurotech in medicine is, why would we even use neurotech in the field of medicine, and what capabilities does this have? So the main goal here is to manipulate the nervous system, and through using specific applications we can do that. So let's get into that by asking the next question, which is, how would we use these devices in medicine? And the main two classes are assessment and intervention. So recording and stimulation have very promising clinical applications. Recording of electron activity includes neuroimaging techniques like EEG and blood flow, which is recorded in fMRI. And there's a lot more imaging techniques, but we've already covered those in lecture. As to intervention, we can stimulate the brain through neuroprosthetic interface, which is essentially a platform for communicating between the nervous system and some neuroprosthetic device. So we create some input-output system and then target the nervous system functions by doing so. And so by doing both assessment and intervention, we're able to control and record some form of brain activity, which is then a very promising application for neurotech in medicine. So specific applications for neurotech in medicine include neuromodulation, DBS, TMS, vagus nerve stimulation, and BCI. And neuromodulation is just the umbrella term for stimulating areas of the brain. And we're going to cover some of the more specific things in this lecture.
All right, so the first technique we're going to go into is deep brain stimulation. And on a basic level, there's three different components. There's the electrode, the extension wire, and the IPG. So the electrode is a thin insulated wire inserted through a small opening in the skull, and it's implanted into a specific area in the brain. The extension wire is also insulated, and it's passed under the skin of the head, neck, and shoulder, connecting the electrode to the internal pulse generator. That's the IPG. The IPG is the third piece of the system, and it's usually implanted under the skin in the upper chest. This image just gives you a better overview of kind of where all of these things are implanted. One of the diseases that deep brain stimulation is used in the medical field to treat is Parkinson's disease. So we're going to go over a little bit about the circuitry and who's affected by Parkinson's disease. So first, a little bit of numbers. So 50 is the average age of disease onset. There's 60,000 cases, like new diagnoses a year, and 1.5 times more men are affected than women. And in the US alone, there's approximately $52 billion in direct and indirect costs in dealing with Parkinson's disease. So symptoms include mild tremors, soft and slurred speech, lack of facial expression, change in gait when walking, stiffness, trouble sleeping, and restless legs. Ultimately, Parkinson's is a disease of the motor system, so it makes sense that the symptoms are predominantly associated with disordered movements. In terms of pathology, the dopaminergic neurons in the nigrostriatal pathway in the basal ganglia die off, decreasing the availability of dopamine in the direct pathway to movement. And of course, we'll go over what all of these terms are in the coming slides. So First, the basal ganglia. So the basal ganglia are a cluster of neurons in the brain named for their location near the base of the brain. The main role of the basal ganglia is to receive inputs from the cortex, process these inputs, and relay an output to the thalamus, which then in turn signals other parts of the cortex. Keep in mind that the thalamus is sort of an information hub in the brain, and so a lot of information passes through the thalamus as it moves from one region to another. The important structures in the basal ganglia include the globus pallidus, which is split into internal and external segments, the substantia nigra pars compacta, and the substantia nigra reticulata, the subthalamic nucleus, and the striatum. The striatum is technically made up of two structures with their own names, but for the sake of this lecture, we just have to know the striatum. And also, for the sake of this lecture, we don't need to know that many specifics about each of the structures. I know the names can be a little bit complicated and intimidating, but just think of them as relay centers or nodes in the overall circuit that allows movement to happen. So like I said before, the actual names don't matter that much, like for us right now. It's how they're connected that matters. So basically, there's two circuits that are theorized to be involved in movement, one in the execution of desired movement and one in the inhibition of undesired movement. The balance between these two pathways is essential to the execution of functional movement. Activation of the direct pathway involves both excitatory and inhibitory signals, but ultimately causes movement. Within this circuit, there is activated activity and constitutive activity. The globus pallidus and the substantia nigra reticulata both constitutively inhibit the thalamus. This means there's continuous activity there. Without this inhibition, the thalamus would constantly stimulate the motor cortex and we wouldn't be able to stop moving. When the cerebral cortex sends a signal to the striatum inhibits the globus pallidus and the substantia nigra reticulata. So as first we have the movement signaled, so that's kind of more of an activated signal, not a constitutive signal, right? So the movement is signaled. We have the cerebral cortex stimulating the striatum, and also it's important to note too, the substantia nigra pars compacta floods the striatum with dopamine, and this pathway between the substantia nigra pars compacta and the striatum is called the nigrostriatal pathway, and that's where all of the dopaminergic neurons die in Parkinson's, which I'll have a better visual of that in the coming slides, but just keep that in mind. So the cerebral cortex stimulates the striatum, the substantia nigra floods the striatum with dopamine, and so then the striatum inhibits the globus pallidus internal and the substantia nigra reticulata, and this inhibition means that the GPI and the SNR will no longer inhibit the thalamus, which is then free to convey excitatory signals to the motor cortex. And so we'll come back to this. Oh, right. I already talked about the substantia nigra pars compacta. So anyway, nigrostriatal pathway, this is the direct pathway of movement, causes movement. So next we have the indirect pathway, which as you can see, it's got a lot going on, but it's fine. We're going to walk through it. So without stimulation, the globus pallidus exterior constitutively inhibits the subthalamic nucleus. The subthalamic nucleus is a glutamatergic excitatory input to the globus pallidus internal and the substantia nigra reticulata, which as we saw in the direct pathway inhibits the thalamus. When the indirect pathway is stimulated, the cerebral cortex directly stimulates the subthalamic nucleus and the striatum. The striatum inhibits the globus pallidus external, which releases the subthalamic nucleus to stimulate the globus pallidus internal and the substantia nigra reticulata, which inhibits the thalamus. Inhibition of the thalamus causes communication with the motor cortex to cease. The substantia nigra pars compacta in this case is active and it inhibits the striatum when the indirect pathway is not supposed to be activated. So again, we have the movement signaled, right? Both the subthalamic nucleus and the striatum are stimulated, and when the striatum stimulates the globus pallidus external, the signal is inhibited to the subthalamic nucleus, which means the subthalamic nucleus can stimulate the globus pallidus internal, which then inhibits the thalamus and stops movement. So basically, what I want you to take away from this is the direct pathway works through stopping the inhibition of the thalamus through the globus pallidus internal, and the indirect pathway works through the excitation of the globus pallidus internal by the subthalamic nucleus. All right. So something to note about these pathways is that the SNPC appears to play a different role in each pathway. We have it's acting excitatory in the direct pathway and inhibitory in the indirect pathway, which can be confusing, but basically the SNPC, so it projects to the striatum, and the striatum contains two types of dopaminergic receptors, D1 and D2. D1 receptors become active in response to excitatory dopaminergic activity, and D2 receptors become active in response to inhibitory signals. And that it's that heterogeneity of the dopaminergic transmission that allows for the regulatory role of dopamine in both pathways. And so, like I said before, it's the neurons in the SNPC that are most affected by Parkinson's disease. This is the nigrostriatal pathway, and it ultimately causes an imbalance between activation of the direct pathway and or activation of the direct and indirect pathways. So the indirect pathway is overactivated, which means we cannot start movements. All of our movements are inhibited, and the direct pathway is underactivated. Okay, so this is just kind of summarizing that. So in the direct pathway, the SNPC neurons die off, which means that the striatum is never stimulated, so it cannot inhibit the GPI and SNR, right, in patients with Parkinson's disease. Now, with deep brain stimulation, we don't exactly know how the mechanism works, but it shows, it's been shown to inhibit the GABAergic projections to the thalamus from the GPI and SNR, which allows for conductions of signals to the motor cortex. So in a sense, when deep brain stimulation is applied, it releases the thalamus, okay? And so, yes, we get movement. Then, in the indirect pathway, so the death of SNPC neurons leads to disinhibition of the striatum, which then inhibits the GPE, causing overactivation of the STN, which then overactivates the GPI and inhibits the thalamus, right? So with deep brain stimulation, it inhibits the activity of the STN, which stops the activation of the GPI and SNR, allowing for activation of the thalamus. So these are all very kind of like hypothetical things. There's still a lot of research to be done because we don't exactly understand these mechanisms, but that's just a pretty brief overview of how that works. So again, deep brain stimulation, when applied to the subthalamic nucleus or the globus pallidus, appears to inhibit the activity of both of those areas, and inhibition of the subthalamic nucleus stops stimulation of the globus pallidus internal, which then releases the thalamus to stimulate the motor cortex. Inhibition of the globus pallidus directly releases the thalamus to stimulate the motor cortex. With that, we will move on to TMS and TDCS.
Awesome. So let's move on to the next techniques, which TMS and TDCS are both non-invasive stimulation methods that are also used in the medical field. So TMS uses a bigger machine than TDCS and involves placing a coil wire on the subject's head. The coil is connected to the stimulation machine by a Vite voltage cable that delivers rapidly changing currents to the patient. In TDCS, subjects are fit with headgear with electrodes. These electrodes are connected by wires to a battery that supplies the direct current. So you can see on this slide, we outlined a few of the differences between these two stimulations. So here we have an image for you guys to visualize what these look like. So essentially what I just described, on the left we have TMS, and you see that there's a large magnetic field above the patient's skull, and then for TDCS, you see that they're fit with a cap, which is electrodes. So the application of this is used in Alzheimer's disease. So let's talk about how Alzheimer's disease is present in our world. So about 1 in 10 Americans over 65 have Alzheimer's. Every 65 seconds, someone in America is diagnosed with Alzheimer's. And $277 billion is the sum of the healthcare costs associated with the Alzheimer's disease in a year in the US. So pharmacological treatments are poorly effective to counteract or delay memory decline. As we see, it's very apparent here. So one of the techniques that we use to inhibit the symptoms of Alzheimer's is rTMS. So repetitive TMS is proven to be beneficial for cognitive functions such as memory recall or recognition in patients with Alzheimer's. A train of pulses is delivered at the same intensity over a time frame, which is thought to increase cortical excitability. The main problem in Alzheimer's is neurodegeneration all over the brain, so increasing excitability could strengthen the neurons and synapses that have not died off and perhaps delay further degeneration. The current treatment of rTMS is applied bilaterally to the dorsolateral prefrontal cortex. The long-term effects of this treatment are unclear, and further research is needed to determine the mechanism through which this treatment appears to help. Another mechanism is TDCS that targets Alzheimer's disease. So there are two ways of altering the pattern of firing in the brain for TDCS, anodal and cathodal. Anodal TDCS depolarizes membrane potential, increasing excitability of neurons in the cortex. However, cathodal TDCS hyperpolarizes the neurons, which decreases cortical excitability. For this reason, we have targeted research into anodal TDCS, and they have found that increasing the excitability of the temporoparietal junction improved word recognition accuracy, and depolarizing the left dorsolateral prefrontal cortex improved visual recognition memory. Of course, this is all very new research with somewhat unclear results, so a lot more work in this field is needed. So if you're interested in learning about the mechanisms behind anodal TDCS, here's just an image of such, but as you can see, it's quite some complicated biology and chemistry mechanisms, so we're not going to get too in depth about that. As for strokes, let's take a look at how this is apparent in our world. So 140,000 people in the US die from a stroke each year. Every 40 seconds, someone in the US has a stroke, and $34 billion is the sum of the healthcare costs associated with strokes in a year in the US. So we can see that this is a very big problem. So the method that we use to target strokes is TDCS. So we know that strokes are caused by oxygen deprivation, which reduces neuronal functioning. So this can lead to motor and cognitive impairments. So increasing synaptic excitability with TDCS can relieve these impairments and improve plasticity in the brain. Some of the proposed mechanisms through which these occur are local changes in ionic concentration, alterations in transmembrane proteins, and electrolysis-related changes in hydrogen ion concentration. Now, how practical is this method, or TMS and TDCS, for treating these neurological diseases? So while they do have very promising futures and we see a lot of potential with them, we come across a few obstacles in applications. So there is a lot of flexibility in protocol, which can be a good thing, but then again, it can lead to lack of standard protocol in numerous studies. So because there is this lack of consistency and consensus within TMS and TDCS studies, which include having small sample sizes and varying protocol setups, this makes it much more difficult to decide on the most effective design for a given experiment. Okay, now we're going to move on to optogenetics, which honestly I find very, very cool and interesting. I've never seen anything like this being done in the neurotech industry. So let's first start off with a common definition of what optogenetics is. So it's a technique that allows genes to be introduced into specific brain cells for further manipulation using lights. The gene codes for light-sensitive proteins. So it was first proposed by Francis Crick, who I'm sure we've all heard that name a few times, who wanted to isolate neurons or neuron clusters to maintain control over. And we couldn't do this with electricity or drugs. So then Carl Dice-Roth in 2005 used light to excite or inhibit neurons and then studied the outcomes of how this portrayed in live free-moving animals. So the way that this works is that green algae are used to create light, or green algae use light to create energy for life. Ion channels are activated with a specific wavelength of light. We know from the microneuroscience lecture that neurons are activated by electrical potentials, and which causes the movement of ions. So this same mechanism happens in these green algae. So we use a common biology technique called recombinant DNA technology, which lets us genetically modify a virus to recombine its DNA with the host cell when injected into the brain tissue to express light-sensitive channels. So the main principles behind optogenetics are the high temporal resolution, which uses pulses of light we can target specific times to, specific spatial targeting, in which we illuminate certain areas of the brain, and the ability to study functions. So genetic modifications are specific to cell type. And the two ion channels. We currently use yellow and blue light to activate these two ion channels, halorhodopsin and channel rhodopsin 2, respectively. So halorhodopsin inhibits neuronal activity, and channel rhodopsin 2 excites neuronal activity. And studies are looking into red light activated ion channels as red light penetrates deeper so we can study deeper brain structures. So here's a really cool application of what optogenetics can do. So the University of Washington and University of Illinois at Urbana-Champaign have used optogenetics to treat bladder pain in mice. And this is the mechanism behind it. So researchers took this light-sensitive ion channel gene and inserted it into a virus, which was done using recombinant DNA technology. These neurons express the gene, while the virus is then injected into the mouse brains. The neuron expresses the gene and creates ion channels in their cell membranes. Then a fiber optic cable and electrodes are inserted near the target cells. We then strike light of a specific wavelength, and the ion channels open, ions flood into the neuron, which can either turn on or off neuronal signals. So the output behavior of the mouse is then recorded in response to this process. So a specific application of this switching off of pain includes this experiment that scientists did, where they targeted nerve cells in the skin that respond to gentle touch, which can also cause neuropathic pain. So scientists discovered a light-sensitive chemical that binds to these skin nerve cells. So a chemical is injected into the affected area, and near-infrared light is illuminated onto this area, causing the nerve cells to withdraw from the surface of the skin, which causes temporary pain relief, because the nerve endings ended up growing back after a few weeks. But a group called EMBL Rome was able to carry out this successfully in mice. So we see a lot of potential with optogenetics. So now, at our very core, of course, UC Berkeley has some contributions to this field. So researchers at Berkeley have developed a holographic brain modulator, and the modulator uses holographic projections to excite or suppress 10 seconds to 1,000 seconds of cortical neurons at once. The modulator can be stimulated up to 50 times per second, mimicking firing rates in human brains. The team working on this developed 3D SHOT, which is the holographic projection system. 3D SHOT stands for three-dimensional scanless holographic optogenetics with temporal focusing. And the goal here is to replace lost sensation due to nerve damage by trying to mimic human brain response to sensation. And this is on the route to developing neural prostheses because it is precise enough to interpret the pattern of activation in the brain. In essence, this technique can allow for talking to the brain, thus recovery lost function. So here's a video of what these researchers were doing.
Okay, I don't think the sound is working.
I don't think there's sound on this video. You can just let it play.
Oh, okay.
You guys can watch the captions. So yeah, that was just a little image of basically what I described in the previous slide.
Okay, so another thing that optogenetics can be used for is studying addiction and other diseases, right? So first, some epidemiology about addiction. As you can see from the graph, alcohol and drug use disorders affect a broad range of people and it costs the economy about $524 billion a year in direct and indirect healthcare costs. So, it's a very problematic issue. Just for some background, addiction is characterized as a psychiatric disorder, which means that it's defined by the DSM-5, which is the baseline text for defining all psychiatric disorders. Generally, there are 4 characteristics that define addiction. Impairment of function without use, recurrent use despite negative social, economic, and physical effects for the user, increased need to take the drug with increased use, and unsuccessful attempts to cease use. These negative behavioral effects are caused by imbalances in the brain that are caused by the drugs, and the severity of the imbalance is dependent on how long a person has been addicted and how much of a drug they take. So as you can see in the fMRI image on the screen, there is decreased brain metabolism in this person who is addicted to cocaine. So obviously very bad effects for all of these things. So we'll go over some basic circuitry. It's centered around what is colloquially known as the reward pathway, which we briefly saw in lecture 3. So the pathway extends from the midbrain through the prefrontal cortex and connects to many structures such as the hippocampus and the amygdala. The structures we'll focus on here are the ones most studied in addiction are called the ventral tegmental area and the nucleus accumbens. The ventral tegmental area is a major cluster of dopaminergic neurons, and it contains inputs from both GABAergic neurons and glutamatergic neurons. The output of the ventral tegmental area is the nucleus accumbens. In most studies of addiction, the level of dopamine in the nucleus accumbens is related to the feelings of euphoria associated with a drug. So increased levels of dopamine in the nucleus accumbens appears to elicit rewarding sensations. So the pathway from the inputs of the VTA, well, that's a ventral tegmental area, I'll just call it VTA because I don't want to say it that many times, to the NAC are messed up when the drugs enter the system, right? So stimulants such as cocaine are known to block the reuptake of dopamine into the presynaptic terminal at the nucleus accumbens, thus increasing the levels of dopamine there. Depressants, such as opioids or heroin, block the GABAergic inputs to the VTA, which allows for increased dopamine release in the VTA, again, raising levels of dopamine in the nucleus accumbens. And so this is just a very surface level peek into what we know about addiction circuitry. But the thing is, there's a lot more going on than just this. A lot of mystery lies behind the inputs to the VTA, right? So the big question mark right there. For example, we know that in cocaine stimulation, there's a change in these glutamatergic receptors called NMDA and AMPA receptors, okay? So the ratio of AMPA receptors to NMDA receptors goes up, which ultimately changes synaptic potentiation. It changes the way the brain makes and maintains connections. So we don't know, however, where these glutamatergic inputs are coming from. And there have been studies that have indicated the prefrontal cortex, which becomes active in drug craving, but no specific circuits have yet to be discovered. So in walks optogenetics, all right? Optogenetics, like we said before, have the advantage of being super specific and very effective. So um, in this study, they used optogenetics to stimulate neurons in the prefrontal cortex, okay? So they basically trained rats to administer themselves cocaine, and of course, some of them began to display cocaine seeking activity. And so when they stimulated prefrontal cortex neurons, it appeared to decrease the cocaine activity seeking in these rats. And this study could be used in models for treating addiction in humans with TMS or TDCS, or just one of the methods we mentioned before. So in the image on the screen, it's a figure from that paper where they talk about stimulation of the prefrontal cortex neurons. So subfigure A, it's just a schematic of when they trained the rats. Subfigure B through E shows the cocaine seeking activity between the two groups, establishing that there's the same activity between the CHR2 groups before turning the light on. And then A through E shows cocaine seeking activity changes with the stimulation of the prefrontal cortex neurons in question. And so, yeah, as you can see, the CHR2 aftershock group displayed an increased latency to seek cocaine as opposed to the ones that weren't tested. So, I mean, in this way, optogenetics was able to identify a circuit that could potentially be used later on in medical treatments of people who are addicted. So finally, we are taking it further.
Right, so I just wanted to emphasize here that while we did cover very specific biological techniques in these methods, there is still so much research to be done in these fields. So especially across all fields. As we emphasized in the first few lectures, we really don't know much about the brain, but these applications that we've discussed are possible with the little that we know. So just imagine how much more is possible as we discover and research more about the human brain. So if anybody's interested in working in the field of neurotechnology, here's just a graphic organizer of the many, many careers available, especially like how widespread these are. You know, you can go into something like rehab for stroke patients or the sport market. So I just wanted to emphasize again that there's so much potential in this field. And as for research, we covered a few of the techniques here like DBS, TMS, TDCS, optogenetics, but there's still so much more being done. And the brain-machine interfaces work with paralyzed patients, which is recovery of lost motor functions. And we're trying to optimize the long-term stability and biocompatibility of brain implants, which is a big obstacle that the neurotech field faces in medicine, as we need to make sure that all of these devices are biocompatible. And on the right here, we've listed some research projects that are being done. So again, we want to thank you for taking the time to listen to what we have to say, and for anybody who's interested, we highly encourage you look into this more.
Yeah, and if you guys have any questions, of course, feel free to email us. I believe there's a discussion page open on the B courses. Also, another quick plug for the survey, we have a feedback form that's also on the B courses. We would really, really appreciate your feedback. And yeah, thanks for sticking with us. It's been fun, everyone.